All right, so thank you, everybody. Today it is my great pleasure to introduce our very own GGI faculty member, Dr. Yan Sui, as the first speaker of our fall lineup. Dr. Sui trained at Peking University and the Chinese Academy of Sciences, where he received undergraduate and graduate degrees in physics, theoretical physics, and biophysics. He completed postdoctoral work at UCLA and Harvard School of Public Health in statistics, biostatistics, and computational biology. In 2002, Dr. Sui joined UT as an assistant professor. He is currently an associate professor in our GGI department, a member of the Institute for the Study of Host Pathogen Interactions, a research member of the Center for Cancer Research, and director of both the Bioinformatics Core and Artificial Intelligence Laboratory of the UT Center for Integrative and Translational Genomics. Suffice it to say, Dr. Sui is the major hub of bioinformatics, machine learning, and computational systems biology research in our department and across the broader UT community. He has led or is currently leading bioinformatics research for over 15 funded projects that's funded by NIH, DOD, AHA, and the VA. And these include currently funded projects for precision medicine, fungal infection, kidney fibrosis, energy metabolism, and breast cancer. Dr. Sui is also committed to developing web databases and services that bring his expertise and bioinformatics tools to a broad research community. Dr. Sui has over 60 publications. Many of these have a lot of citations and are very influential. And his lab has developed over 10 software systems that are useful to address a range of bioinformatics problems, from Bayesian network modeling to variant impact predictions. Many of these are freely available at compbio.uthsc.edu. So Dr. Sui, thank you for kicking off the GGI Fall Seminar Series with your most recent and exciting research on machine learning, health disparities, and more.
Well, thank you for the introduction. The first part of my talk is about machine learning for health equity. So omics data is essential for precision medicine. However, over 80% of GWAS and clinical omics data were collected from individuals of European ancestry, which only comprise about 16% of the world's population. So there is a severe data disadvantage for the vast majority of the world's population. The figure on the left is from the GWAS diversity monitor. Here we can see that over 88% of all the participants of the genome-wide association studies are of European descent. And the diversity was not improved in the recent years. From the figure on the right, we can see that the similar data inequality in some of the largest public clinical omics data sets. So this kind of ethnic composition is typical in current clinical omics data sets. In many studies, people have observed ancestry-associated DNA mutations, epigenetic modifications, and RNA and protein expression. And ethnicity-specific reference intervals for clinical lab tests have also been observed from electronic health record data. This indicates that data distribution differences may exist among the ethnic groups, which has important implications for machine learning with multi-ethnic data. Currently, the data foundation for building AI-powered precision medicine is imbalanced for the lack of adequate data for non-European populations. We use machine learning experiments to answer two questions. Will the data inequality lead to AI models with low performance for the data disadvantaged populations? And if so, how to improve AI model performance for these populations? For our machine learning experiments, we assembled tasks and data sets using the data from the Cancer Genome Atlas, or TCGA. TCGA dataset contains multi-omics data with the matched cancer outcomes of about 11,000 patients from multiple ethnic groups. The data advantage of ethnic minority groups exists across almost all cancer types in TCGA. So it provides an excellent test bed for the comparison of multi-ethnic machine learning schemes. The machine learning tasks for our experiments are to predict the clinical outcomes from omics data. We want to distinguish between individuals with good and bad clinical outcomes. We used 4 types of clinical outcomes as prediction target variables: overall survival, disease-specific survival, disease-free interval, progression-free interval. Currently, the standard multi-ethnic machine learning scheme is to train a model using the mixture data from all ethnic groups, regardless of the cohort's ethnic composition. A not so frequently used alternative multi-ethnic machine learning scheme is to train a model for each ethnic group separately using its own data. We call this scheme independent learning. And we compare the mixture learning and independent learning schemes using the machine learning tasks assembled from TCGA data. For example, this machine learning task is to predict if the glioma patient's overall survival time is longer than 3 years. The dataset contains protein expression data from 22 African American patients and 315 European American patients. The performance of mixture learning model looks great. The AUROC, or area under ROC curve, is 0.88. Unfortunately, almost all machine learning works that use multi-ethnic data stopped at this step. However, if we further test the model performance on EA and AA patients separately, we can find that there is a large model performance gap. While the model performance on the EA group is high, its performance on the AA group is much lower. So clearly the overall high performance from mixture learning is driven by the high performance on the EA group. The mixture learning scheme, which is a current prevalent learning scheme for multi-ethnic machine learning, may lead to unnoticed model performance disparity gaps if we do not test.
Yan, this is Andy Griffith. I'm not familiar with this graph on the right, and I wondered if you could just kind of explain what the ROC curve is for us novices. Thanks.
So our ROC curve is a curve that this is people use that for the evaluation of machine learning performance we use the area under ROC curve to evaluate if machine learning model perform well or not. The best performance, the perfect prediction would give you AORC of 1. So if you got AORC equals to 1, then you have the perfect performance. And for complete random predictor, the area under ROC curve would be 0.5.
That's helpful. What does ROC stand for? Maybe you said this and I missed it.
So that has it's if I remember that correctly, that's called the receiver operating characteristic. So that's from I think radiation medicine. I'm not sure, but that's it has a long history but just call it ROC curve. So yeah. That's a metric people use to evaluate machine learning model.
Okay, thanks. Now I don't feel so foolish asking.
That's a good question.
World War II, though, so they had to figure these out for gunshots and dropping bombs and things.
Okay.
Yeah, I also have a question. This is how. So in your EA, in your mixture learning, the EA plus AA is 0.AA, and EA is 0.89, and AA is 0.7. Is this, is the overall mixture 0 close to EA because you have a lot more EA sample than AA sample?
Right, that's right.
Okay.
So in the training data, you have like over 90% of the cases are EA patients. So the performance was driven by the performance on the EA patients.
Okay, got it. Thank
you. It's a performance of the independent learning model for the AA group. It's much lower compared with the performance of the independent model for the EA group.
And that is also because you have a lot less AA samples, right?
Yes, that's because the training data for the AA group is inadequate.
Okay.
So both mixture and the independent learning schemes generate significant performance gaps between the two ethnic groups. We found that transfer learning can improve machine learning model performance for the data disadvantaged ethnic groups. In transfer learning, knowledge learned from the source domain, where training data is abundant, can be transferred to assist the learning tasks in the target domain, where training data is inadequate. So the knowledge we talk about here is not the explicit knowledge that can be expressed using mathematical language or a natural language. The knowledge is we talk about here is encoded in machine learning model, for example, in the structure and parameter of a neural network. In the glioma prognosis task, transfer learning improved the machine learning model performance for the AA group. Transfer learning reduced the model performance disparity gap from the mixture learning by 42% and reduced the gap from independent learning by 68%.
Yan, sorry to interrupt you, but there is like one question in the chat maybe from Dr. Pfeffer that relates to the last slide. Is there a difference in OS and EA versus AA and glioma by this model?
Let me see, is that in OS, in EA versus A?
I think in the previous slide.
Yeah, I'm just asking what is the slide showing? Is it showing you that there's a difference in overall survival? Because I'm not understanding. This is Larry Pfeffer.
Oh, this is showing you the accuracy of the prediction. So this is not. This does not mean that AA patients have worse survival, overall survival compared with EA patient. This is just the prediction accuracy for the AA patient is lower compared with the prediction accuracy for the EA patients because of the data inequality.
So one thing that wasn't clear to me that may be helpful for people. So the machine learning task, so I know that you had the protein and the transcript data in there. So is this looking at whether or not the machine algorithm can appropriately diagnose the positive or negative outcome based on markers, the transcript or protein markers, and given the like survival time? Is that what's?
The survival time would be the prediction target. So we use protein data, protein expression data, and mRNA expression data to predict the overall survival. So we use lots of features, hundreds of features. It's not like we just make the prediction based on a few markers. Okay, let's. And these are the deep learning and transfer learning models we used. The technical details of the models are described in this paper. Yan Gao, a postdoc working with me, did all the machine learning experiments in this study. And these are the results of our machine learning experiments on some other cancer types, omics data types, and clinical outcomes. And here are more results on the comparison of the multi-ethnic machine learning schemes. There is a common performance pattern. You can see there's a common performance pattern, which is characterized by model performance gaps between the ethnic groups from the mixture and independent learning schemes, and also by transfer learning improving the performance of for the this data disadvantaged ethnic group. So we performed the machine learning experiments on 224 tasks to predict cancer outcomes using mRNA and protein expression data from EA and AA patients. A total of 21 types of cancers and 4 clinical outcomes and 4 clinical outcome endpoints were involved in this learning tasks. And this figure shows the degree of data inequality in the learning tasks. The percentage of AA patients ranged from 6% to 25% in these 224 learning tasks, with a median of 12%. The median percentage of mRNA or protein features differentially expressed between the 2 ethnic groups in the 224 learning tasks was 10%. So the degree of differential expression is related to the degree of data distribution difference. Among the 224 learning tasks, 142 have a performance gap larger than 0.05 AURC, and 88.7% of these performance gaps were reduced by transfer learning. So overall, the performance gap from mixture learning and independent learning schemes are statistically significant. The model performance improvement for the data disadvantaged AA group by transfer learning is also statistically significant. So then we want to identify the key factors underlying the disparities in machine learning model performance. We hypothesize that the data inequality and the data distribution discrepancy are the key factors underlying the machine learning model performance disparity between ethnic groups. And both factors can be addressed by transfer learning. We use synthetic data to test this hypothesis. We develop a simulation model to generate synthetic data. The parameters of the synthetic model represent these two key factors, data inequality and data distribution discrepancy between ethnic groups. And using synthetic data, we can control the presence and absence of the two key factors in the synthetic data generating process. So we can study the effect of all the four possible combinations of presence and absence of the two key factors in the multi-ethnic machine learning schemes. Synthetic data 1 was generated using parameters estimated from a real data learning task. The performance pattern from synthetic data 1 is very similar to the performance pattern from the real data learning task. We can see that both have model performance gaps from the mixture and independent learning schemes, and transfer learning improved the model performance for the minority group. So this
can you explain what is data inequality and distribution discrepancy, please?
Data inequality means so there are more data, much more data for one ethnic group, and for other ethnic group, there is much less data. So that's a data inequality. And data distribution discrepancy means the difference between the distributions of the features used for training. So, does that answer your question?
So data inequality basically is sample size discrepancies between the two populations.
Actually you can the degree of data inequality can be measured by the ethnic composition of the cohort, right? So if you have 90% of the cohort are European Americans and 10% of the cohort are African Americans, then this is it. You have 90% of the data from the EA group and 10% of data from the A group. So this is called data inequality.
Okay.
Okay, let's continue. So we can see this both they both have a model performance gap from a mixture and independent learning scheme, and the transfer learning improved the model performance for the minority group. And this indicates The simulation model captures the key factors underlying the performance pattern. Synthetic data 2 has data inequality, but no distribution difference between the two ethnic groups. For this synthetic data, there is no performance gap from mixture learning. However, the performance gap from independent learning remains. The synthetic 3 has equal number of cases from the two ethnic groups. So there is no data inequality, but it has a distribution difference between the two ethnic groups. Synthetic data 4 has no data inequality and no distribution difference between the two ethnic groups. For these two synthetic data, there is no significant performance gap from either mixture learning or independent learning. For all the synthetic data sets, transfer learning achieved best performance for the minority group. The machine learning experiments on synthetic data show that the performance gap from mixture learning is caused by both data inequality and data distribution discrepancy between ethnic groups. While the performance gap from independent learning is caused by inadequate data for the minority ethnic group. And transfer learning can reduce these performance gaps. So here are some key messages for this part of the talk. Biological data disadvantage is a health risk factor for the vast majority of the world population. AI greatly empowers precision medicine, but in the meantime, opens up a major pathway for this risk factor to exert its effect. AI-powered precision medicine will be less precise for the data disadvantaged populations, which will produce a new type of health disparity. The new health disparity can impact any disease where data inequality exists. Their negative impacts would be broad. And using transfer learning can reduce the negative impact of data inequality, thereby mitigating the health disparities arising from data inequality. So we hope our work will provide a starting point for a new paradigm for multi-ethnic machine learning that implements regular tests of the performance of machine learning models on all ethnic groups to identify model performance disparities and use transfer learning or other techniques to reduce performance disparities. This is essential for reducing health disparities arising from the long-standing biomedical data inequality among ethnic groups.
Can I ask a question, Yan, before you move on?
Sure.
This is Andy again. I was reading, I think it was in Nature this week, there was a one-page editorial written by a population geneticist at Stanford on the use of race and ethnicity in genetics. And, you know, some of the kind of things we've heard before about how there's not really a biologic construct under these, but brought up the really good point that, you know, we're really, the global population is evolving from, you know, a good number of kind of ethnically and reproductively isolated groups, or another way to put it is kind of mono-ethnic groups, into a melting pot of people of all sorts of different races and ethnicities. And, you know, in many places in the US, it's more common to have patients who identify as multiracial than not. And what I'm getting at is I'm just thinking about how we're going to deal with that moving forward.
You mean how we deal with that?
Right.
In machine learning? Or
you know, so let me put it to you another way. So to implement machine learning in the clinic as, you know, a decision-making tool, do you think in the future, are we going to have to be able to plug in the ethnicity of a person? And what if it's unclear or multiracial or we don't know? And I'm not trying to, I think this is a great tool. I'm just wondering if you're thinking one step ahead here about how we can overcome this problem of ethnicity as a construct, because it's becoming less and less useful. Maybe this is just something to think about, and I don't know if there's an answer. It's just something that a lot of us are thinking about as the population becomes more mixed.
I think this is a really complex question, and there's no simple answer to this question. So people argue about if ethnicity should be used in medicine at all, or but in our case, we actually use that. We don't use that as an input for the machine learning model. We just train different machine learning model for different ethnic groups, because for some ethnic group, there's simply no adequate data. So that's the question we want to solve. And so the question you ask is much more complex. I don't know.
Okay. Sorry to slow you down.
Well, that's an interesting question. So in the second part of the seminar, I will talk about causal inference in system genetics. A major goal of system genetics is to elucidate the molecular networks that translate DNA sequences into phenotypes. Genomic variations can cause variations in transcriptome, proteome, metabolome, and the phenome. So we want to discover the causal pathways connecting genome and phenome. Bayesian network is a statistical model that, a statistical method that can be used for causal inference. You can learn the Bayesian network structure and the parameters from data, and then use the Bayesian network model to make predictions. And deseparation is the most important concept in causal inference with the Bayesian network. It provides the connection between the causal claims expressed in the directed graph and the statistical model of data. We can use directed graph to express to represent causal relations between variables. For example, this directed graph has 4 nodes. Each node represents a variable. Here, A and B are the causes of C, and C is the cause of D. The causal relations between the variables can be expressed in terms of independent relations based on deseparation. Deseparation gives the necessary and sufficient conditions for 2 sets of variables to be probabilistically independent, conditioned on some other variables. For example, this A and B are unconditionally independent here. If the causal model is true, we should observe the corresponding probabilistic independence from the data. So this is another independence relation in this directed graph. D is independent from A and B conditioned on C. And this is a corresponding probabilistic independence that we should observe from the data if the causal model is true. So so in this way, we can use the data to test the causal model. Here is an example of causal inference with biological data. In this study, we use omics data from the BXD mice infected by Chlamydia psittaci, a type of bacteria that can cause life-threatening zoonosis. BXD is a set of recombinant inbred strains derived from two ancestor strains, Black 6 and DBA2. The BXD genomes contains about 6 million DNA variations and have been widely used for QTL mapping. A QTL is a genomic region associated with a quantitative trait or phenotype. We found a QTL on chromosome 1 regulating several cytokines. We also found a QTL on chromosome 11 regulating the weight loss after infection and neutrophils, macrophage activation status, and chlamydia load. We then constructed a correlation network. The network has two parts. A large subnetwork contains a QTL on chromosome 1 and the cytokines. And a smaller subnetwork that contains the QTL on chromosome 11 and its associated phenotypes. Weight loss is used as a measure of disease severity in this study. So this small subnetwork is more directly relevant to disease severity. And as you know, correlations does not imply causality. So we learned a Bayesian network model that contains the QTL on chromosome 11 and its associated phenotypes. Here, the numbers are the posterior probabilities or confidence levels for the directed edges. With this Bayesian network model, we can use the do calculus to make predictions on the effects of experimental interventions. The do calculus allows us to compute the post-intervention probability distributions for the variables affected by the experimental interventions. So we use this BM model to predict the effect of macrophage depletion in B6 and D2 mice. And our collaborator Isao Miyuri did experiments to test the predictions. The Bayesian network model predicted that macrophage depletion would cause the B6 mice to lose more weight, but have no effect on the weight loss in D2 mice. So these two predictions were consistent with the experimental results. The Bayesian network model also predicted that the macrophage depletion would cause neutrophil levels to increase in both B6 and the D2 mice. And these predictions were also consistent with the experiments. Finally, the Bayesian network model predicted that macrophage depletion would cause the pathogen load to increase in both Black 6 and the DBA2 mice. The experiment showed that the macrophage depletion did cause an increase of pathogen load in D2 mice, but not in Black 6 mice. The pathogen load was actually decreased by macrophage depletion in B6 mice. So 5 of the 6 predictions were consistent with the experimental results. This kind of prediction is difficult because the training data does not include information on macrophage depletion. The training data was collected from mice infected by chlamydia. Those mice had a normal macrophage. The Bayesian network can learn from observational data and predict the consequence of experimental intervention. This is the most powerful feature of causal inference, in my opinion. So we developed the Bayesian network web server for interactive causal inference. It is a web-based network modeling environment that integrates algorithms for probabilistic causal inference using Bayesian network. Jesse Zervos, our postdoc working with me, is the lead developer of the Bayesian network web server. The Bayesian network web server includes an interactive user interface for adding structural constraints to the Bayesian network. This would allow you to incorporate biological knowledge into Bayesian network modeling. This feature is particularly useful for multi-level omics data. You can assign each level of omics variables to a specific tier. For example, you can assign the genotype node to tier 1, molecular trait node to tier 2, and disease phenotype nodes to tier 3. And then you can define the relations between the tiers. And you can also require or ban any specific edges, between any pair of variables. This is an example of using the Bayesian network web server to model yeast gene networks. In this study, we used genotype and the mRNA expression data of 1012 yeast strains. We identified an EQTL hotspot on chromosome 7. We then learned a Bayesian network model that includes the EQTL and 8 genes regulated by this EQTL. In this Bayesian network model, AFT1 is a known transcription factor, and there are several known regulatory relations labeled by green stars. Other regulatory relations in the model needs experimental validation. If anyone is interested in test such models experimentally, we will be glad to provide some high confidence candidates. We also have this kind of model for mice. And this is another example of using Bayesian network web server to construct the causal network of clinical factors determining the weight gain after kidney transplantation. here are some more application cases of the Bayesian network web server. It has been used in a wide range of studies, from the influenza disease severity mechanism to fungi development and evolution and various diseases. And of course, the application of Bayesian networks is far beyond biology and medicine. You don't have to have biomedical data to in order to use the Bayesian network web server. And in the last part of the talk, I will briefly introduce 3 databases for non-coding genomics. As you know, microarrays are a class of small non-coding regulatory arrays. Microarrays can cause mRNA degradation or translation inhibition by binding to the target mRNAs. Genetic variations in microarray seed region or binding sites can disrupt the interaction between the microarray and the mRNA, rewire the microarray regulatory networks, and cause diseases. We developed a database called polymers. Here, polymers means polymorphism in microarray and their target sites. The database contains a large number of genetic variations in microarray seed regions and microarray target sites. It also provides an integrated platform for functional analysis of this kind of genetic variations. Microarrays can also bind to other non-coding arrays. Many types of arrays, including mRNAs, circular arrays, and long non-coding arrays, may compete for the binding of microarrays. These arrays are called competing endogenous arrays or CE arrays. The CE arrays can indirectly regulate the abundance of a microarray targeting mRNA by competing for the microarray binding. And the aberrant microarray CERA interactions are associated with many types of cancers. So we developed SOMA-MIR, a database of cancer somatic mutations that potentially alter microarray CERA interactions. The SOMA-MIR database also provides an integrated environment to analyze these microarray CERA related somatic mutations in the context of biological pathways and other biological knowledge. The third database I will introduce is the CTCF binding site database. The CTCF protein functions as a genomic insulator, which set boundaries to block the interactions between enhancers and promoters. CTCF is also important for the three-dimensional organization of the genome. We developed the CTCF binding site database, which is a collection of known CTCF binding sites And the database also provides tools for the prediction and functional analysis of the CTCF binding sites. And India Bhattacharya, a former postdoc in my lab, is the lead developer of these 3 databases. These bioinformatic resources are widely used. Our web server has more than 200,000 visits each year from about 5,000 sites worldwide. And here are the web addresses. Welcome to try them out. So finally, I want to thank the current and former members of my group and our collaborators. And thank you for your attention. I'm glad to answer questions you might have.
If no one has a question, I'll start while people are thinking about additional questions. So great talk. I was really interested in the machine learning aspect of the talk. I was noticing that in the transfer learning method for the machine learning task that was applied to the African Americans, there was a big spread in the accuracy of those tasks.
Yeah.
So I'm wondering if
that's a great observation. The inadequate training data not only lead to low performance or low accuracy, it also lead to the unstable results. So you have unstable predictions when you have a very small amount, only have small amount of data.
Okay, I was wondering if it was because of that lack of data, or if there was something about the cancer types or the protein or transcript data that separated those.
That's actually universal. That's caused by the inadequate training data. It does not depend on specific cancer type and so on.
So if you see that kind of spread, that's really an indicator.
Yes.
Okay. Thank you.
Yeah, I also have a question on transfer learning. So what is the minimum size of the minority group that you can deal with practically to get a good transfer learning result?
There's no definite size that you
but like when you have maybe I mean,
so
in your transfer learning model examples, what are the typical size of your minority group?
I think it ranges from like 20 something to 100.
Okay, so that's relatively small.
Yeah, relatively small.
Great, thank you.
Graduate students, any questions?
That was a really great talk.
Yeah, I think one last question. I mean, maybe you can give some advice, because so more and more people are using machine learning approaches. When you read papers, if you have to review papers, maybe you could offer a few pointers on when you're looking at these things, what you should be alarmed by when you see these types of machine learning papers. Because I know.
Yeah. So machine learning are increasingly used in biomedical research, and so we will talk about machine learning. Machine learning is a big field. It includes a lot of method, but most exciting recent progress are all from a branch called deep learning. So we use deep neural network to it enables a lot of things like self-driving cars and expert level image analysis. I mean, biomedical image analysis, and it also can beat some of the world's best game players. And it very recently, and the DeepMind, a company, a UK company, I think that's a Google company actually. They just published a paper in Nature. It's called Alpha. They developed a method called AlphaFold, and it greatly improves the prediction accuracy for protein structure prediction. So so there are a lot of very exciting progress or breakthroughs in just in the past few years.
So that's an interest. I noticed that too, and there was. This has been a huge, sort of a holy grail in understanding biology is going from primary sequence to structure. And a month or few ago, there was also a release, and I don't know if it in the press about this. And I bring it up because I was really interested. And then Rob told me that it's really not working. And so I know this is a bit of an aside, but I'm curious because when I read the results of that study, it looked like, they were able to generate all sorts of data. And I guess the question is, why is anyone questioning its validity? And I'm also wondering, how in general we might go about assessing the significance of machine learning, because one thing I noticed in your presentation and others is the statistics we use to assess significance aren't always the same, are they, than what we use for other kinds of data sets? I'm really naive about that, and I'm curious.
So when you evaluate the performance of machine learning models, we sometimes use multiple independent runs to assess if the result is robust, and or if the result is stable. And then we end up with the kind of a box plot, right? Just like the ones I showed. And we just use the statistical tests to calculate the p-value and see if the difference between two models, difference between the performance of two models is significantly different.
So that's where I'm curious, because when the world got into GWAS, the p-values meant something very, we had to change how we defined, a p-value. And now, of course, there's a lot of geneticists or statisticians on the Zoom meeting. I mean, some people don't even think we should be using p-values to measure significance. So I guess my question is, with machine learning, do we use the same old kind of measures of significance that we always did? So if it was a p-value, I know people don't want to use 0.05 anymore. They talk about using 0.01. I mean, where's the field at? What is significant?
Right, you mentioned GWAS. In GWAS, you have multiple testing problem because you have tens of thousands of markers. You need to do tens of thousands of statistical tests. So that's so people said the threshold for statistical significance very stringent, and but here we just try to just compare the two models' performance, and we probably have 10 or 20 independent runs. So this is not a multiple testing problem. So we don't have to control p-value like we do for GWAS.
I see, and that makes sense. I guess I was thinking with machine learning, you've got so many iterations with so much data that created kind of it's not a multiple testing problem. So thanks for clarifying that.
But I think that does raise the issue of how robust the model is, whether it's overfit, and whether it's going to generalize to another population, which is obviously something that Yan has dealt with very explicitly, since he's trying to figure out what he can transfer from a West Eurasian population to an African population or an African American population. So it's that ability to accommodate a new group of individuals and generate accurate predictions, which is your measure of the robustness. So if Yan claims that his ROC curve now is yay good with the population that he used for the training, after the transfer, before the transfer, the question is now if you throw 10,000 more subjects at him that have been categorized by professionals, can he do as well as the professionals with the machine learning method? And
this is a transferability problem, I think. So you have to have a model. You will always want to test if the model is valid for another group, for another data set. Sometimes different data sets may have different distributions. So people call this data set shift or data set shift. So you have different distributions from different data sets. Then the transferability of your model need to be tested. And transfer learning can actually improve the transferability.
Yeah. I have a couple of comments too, on the same line. So this is Siamaka speaking.
Mm-hmm.
So first, I think it's very important to know that statistical significance doesn't mean any clinical relevance or clinical significance. So this is important and also related to Megan's question as well. So this is very important for us to know that, and also on the same line with Dr. Griffith's comment that p-value currently is just very, sometimes maybe misleading. For example, if you have thousands of samples, assume that in the case of glaucoma, we have the IOP for two groups, that when we have a lot of number of samples, then very easily the p-value is going to be significant between these two groups. And for example, one has 1 millimeter of mercury of IOP greater than the other group, but this is not clinically significant actually, although it is based on the p-value. Maybe the p-value is a very small number, and the reason is that because we have a very large number of samples. So p-value was designed previously to work with statistical models that typically work with the number of samples in the range of 100 or maybe 50 or maybe just less, but now we have thousands of samples. Two groups, the IOP difference is absolutely from the statistical significance. It is significant, but clinically doesn't have any meaning over here. So very important to just note to these, a very critical differences between these points when we are dealing with big data and also with machine learning. And also another point is that empirical validation is totally different from clinical validation. Most of the models currently are empirically validated, not based on the clinical. And even sometimes the outcome that we are measuring has no association with the endpoint. For example, in glaucoma, the endpoint is the vision. I mean, ultimately, we have to check the vision of the patient. No matter what is the IOP, no matter what the structure of glaucoma, for example, optic disc look like. Maybe it looks like glaucoma or suspicious, but at the end, the patient is just seeing, nicely, and there's no problem with the vision. So this is also very important to just make sure first that there is a very strong association between what we are measuring, or we are just developing the model, the deep learning model, or whatever type of machine learning model, with the endpoint in clinic. So these are very important points that should be considered. And also, as you also mentioned, a number of examples that maybe a model that is working, in somewhere with a data set that is sitting somewhere works fine, but it doesn't work on the other data sets. It could be due to a number of reasons. It could be due to the model, the limitations in the model, the limitations in the data set. And also, as I said, we have validated the model just empirically, not based on the clinical true clinical situation. And that's why now currently FDA just mandates 3 steps of validation. One is just first making sure that there's an association between the outcome and what we are measuring, and then empirical validation based on just the data that you have, and then ultimately based on the clinical data. And so these are, very important things that are related to the question that Megan also just posed, that these are the things that we just want to review a paper, or just study machine learning model, just take into account.
This is Andy again. So CMAC, thanks for that explanation. You mentioned the FDA and kind of what they require, and are those requirements you talked about specific for machine learning implemented as a diagnostic tool?
Okay, so those are actually for software as a medical device. They call it SAMD. But and the machine learning models and deep learning models are part of that. So yes, it's just like an umbrella that just covers the machine learning and deep learning models as well.
So I had a question too, and I asked this at a presentation this week that Bob Davis and Rob gave to otolaryngology, actually. And, as a clinician, if we're presented with a new tool for diagnosis or detection, we're used to thinking about the sensitivity and the specificity of that test. And we're not familiar kind of with the way you're measuring performance of machine learning. And of course, clinicians, their eyes are going to roll up into their head if you present them with something they didn't learn in medical school. So I guess my question is, at what point do you, to encourage uptake of these technologies, isn't that what you want to do? Like kind of test it out on a, on a data set, but interpret in terms of sensitivity and specificity for detecting or predicting what you're doing? Where's the field at in terms of that? Or am I barking up the wrong tree?
Actually, that's a great point. So based on, I think, different applications, for example, if you're just dealing with a screening, and it's the screening of glaucoma, for example, the prevalence is pretty much low. So you typically need a very just highly accurate model that can be used effectively for a screening. These numbers, the sensitivity and the specificity, they have different interpretation based on different applications. So for low prevalence diseases, we need a very high accurate, and we have to work on predictive probability compared to sensitivity and specificity alone to just develop a model that is going to be used effectively for those diseases. But for some of the diseases that are more prevalent, again, in ophthalmology, maybe cataract, for example, maybe the models are fine. With 95% specificity or just over 80% sensitivity, I mean, yeah, for detecting those, it is possible to use those. But again, metrics also very important, which metric we use for which type of application. If it is a screening and if the disease is low prevalence or high prevalence, we are going to use different metrics. Typically, the people are now just working on ROC. ROC is very general, and also we have to be very careful about that as well. When the data is highly imbalanced, means that we have a large number of normals and then a very small number of abnormals. That is the case for most of the diseases and for most of the biomedical problems. Then the ROC is not a good representation. And we have to make sure that we use, a number of different metrics to show how the model is working.
Thanks a lot. Sorry to segue, but it's not often I get so many smart people together in one place to ask my questions.
That's great discussion.
All right, yeah, that was a very enlightening discussion. Very helpful comments for, trying to deal with reading and understanding machine learning. And now, Yan, of course, we know to go to you for help, so. All right, well, thank you, everybody. And thank you, Yan. I hope you guys all have a great weekend.
Thanks a lot. That was a
Bye.
great seminar. Thanks.
Thank you.